In this video, we are going to summarize the treasure hunt lab, which is the lab we do in Honors Biology for Unit 8, which covers biotechnology. In this particular lab, the scenario is that a treasure chest has been found in the creek at Pope High School, and we are trying to figure out who does that treasure chest belong to. Now, thankfully, the treasure chest had traces of DNA on it, so we have actual DNA evidence that we are comparing to all of the Honors Biology students at Pope to see if any of those students were the rightful owner of the treasure chest. Now, in order to do this, there are three main tasks that we have to complete. The first one is we actually need to extract the DNA from all of the Honors Biology students at Pope. And this was done in the very first part of the lab when you used a toothpick to gently scrape your cheek cells and put it into an extraction buffer, and then you heated it up to 95 degrees Celsius. That allowed the membranes to open up and allowed your DNA to be released from your cheek cells, and that DNA is then used in step 2. So the DNA is copied using the process of PCR, which stands for polymerase chain reaction. And then the copied DNA, if we move on to step 3, what you did was you put that DNA into a gel, and we used gel electrophoresis to create a DNA fingerprint. And the DNA fingerprint is the unique pattern of DNA that each person has based on the alleles they have inherited. So to understand what that DNA is, we need to know a little background about VNTRs. VNTR just stands for variable number of tandem repeats. Variable number means different people have different numbers of these repeats, and then the tandem repeat means that there are sections of DNA that are repeated together or in tandem, and that's where our abbreviation comes from. Now, the thing about these VNTRs is they are found in the non-coding regions of DNA. Non-coding simply means that it's the DNA found in an area that's not being used to make a protein. So, as mutations naturally occur over time in the non-coding regions, they have no impact on a protein, so they don't actually hurt the individual, so they tend to accumulate over the years. What we are doing is we are looking at a particular region called the D1S80 region of chromosome 1, and we have PCR primers that allow us to copy just this region right here of the chromosome. So we're not copying the entire chromosome number 1 for everybody. We are just looking at this region here called the D1S80 locus, and in this location on the chromosome, there is a variable number of repeats. Some people might have 20 repeats in this area, you might have 34, you might have 2. It just depends on what you have inherited from your mother and what you have inherited from your father. So you most likely have 2 different versions of this D1S80. One might have fewer repeats than the other, as you see in this picture. Now, when we're looking at the repeats, this particular section we're looking at is 16 base pairs. What that means is each of these little rectangles would represent a pair of bases. So if there's an A on one side, there's a T on the complementary strand on the opposite side. So 16 of those pairs is repeated, and that's what we mean by 16 base pair, or BP. So let's think about that PCR tube that you created. In order to copy DNA, you need to put everything in the tube that's needed to make the copy. So obviously, one of the main things you need is your template DNA, this strand of DNA that was extracted from your cheek cells. The template is what is being copied. We also need primers. There's one on either side, so this is just calling them forward and reverse. And remember, the primers we chose are specific to the D1S80 VNTR region. And then you also added something called Easy PCR Master Mix. The master mix is a mix of solutions that has everything that the reaction needs, like the DNA polymerase. Remember, DNA polymerase is responsible for making that new DNA. It has nucleotides in it, just free-floating A's, T's, C's, and G's. If you're building DNA, you obviously need the building blocks for that. So all of these nucleotides down here are just free-floating, ready to be used to make your complementary DNA. And then the last thing that the master mix has in it, it's just a solution of ions and things that help stabilize the reaction. We want the reaction to be completed with accuracy, so scientists have optimized a solution to allow that reaction to take place. So remember, and we talked about this previously in our class notes, that PCR is simply a series of cycles. We used 32 cycles in our PCR for this lab. And regardless of the type of machine, your class may have used the mini PCR machine or this larger thermal cycler here. They both do the same thing. And these are the basic steps here. So you have DNA at the beginning, double-stranded, normal 5' 3' anti-parallel DNA strand. What the PCR machine does is a series of 3 steps, and we call those 3 steps one cycle, and that is what in our lab we did 32 cycles of these 3 steps. So the first step is denaturation. You can see in the picture here the 2 strands have separated from each other. So that's what occurs during the denaturation step. Then we cool it down to anneal the primers or attach the primers. So you can see this primer is attaching here, this primer is attaching here. They can only attach if the temperature is lowered. So that's why step 2 has a lower temperature. And then step 3, our extension step, we raise the temperature a little bit, not quite as high as we were at the beginning, and that allows this DNA polymerase to create the DNA copy. So these 2 strands of DNA you see are in the process of being created. So at the end of these 3 steps, we now have 2 pieces of double-stranded DNA, whereas we started with only 1. And all of this is just one cycle. Again, we did this 32 times, essentially doubling the amount of DNA every single time we went through a cycle of these 3 steps. So now that we have copies of DNA, the next thing that you had to do was your gel electrophoresis. And in order to do gel electrophoresis, which again separates DNA fragments by size, you had to load your sample into a gel, turn on that gel machine, and then the electrical current separated the pieces of DNA. If you need to review that, you can go back and look at your gel electrophoresis worksheet or the notes that we took over the tools of biotechnology. That process is discussed in greater detail in both of those places. For this lab, what we need to focus on is the ladder. The ladder was a DNA sample that was loaded in your gel in the very first lane, and it consists of DNA that is known fragment sizes. So all of these bands that you see here, scientists know exactly how big the DNA fragment is that makes this band because that's how the DNA ladder is created. We know that the band that shows up closest to the top is 1,000 base pair. The one that's closest to the bottom is 100 base pair. So if you compare your DNA bands to the bands in the ladder, you can estimate the approximate size of the fragment. Now it's important to know that the DNA in our VNTR region, the repeated section actually has an extra 180 base pair on either side of it. That's just the region where the primers are going to be binding. And so we have to take that into account. This extra 180 base pair is included in whatever size we estimate by looking at the gel. So we have to take this 180 base pair into account when we are trying to calculate how many repeats are in your VNTR. Now remember that VNTR had 16 base pair. So once you get the size of your fragment and you subtract the 180 base pair that are the flanking region, whatever's left, you can just divide that by 16, and that will tell you how many repeated sections of 16 nucleotides you have. So for an example here of exactly how you do that, here is our ladder shown on the left. Here is a student's DNA fragment. This represents one of the alleles that that student would have inherited. We can look at this diagram and pretty easily tell that that DNA fragment is approximately 400 base pair because it lines up with the 400 base pair band on our ladder. So what we know is 400 base pair nucleotides make up this piece of DNA. We have to take into account the 180. All of this stuff here and this stuff here where the primer is binding, we have to subtract that out because it's not a part of the actual repeated region. The VNTR region is here in the middle. So we subtract the 180 and that leaves us with the number of repeats that would be in this section here. So whatever is left in this section, if we divide that by 16, in this example, we're going to get 14 repeats. So again, use your ladder to estimate the size of the fragment. Once you have the size of the fragment, you have to subtract the 180 base pair that would represent this flanking region. And then you divide that by 16 because you know there are 16 base pairs in every single repeat. That will tell you how many repeats you have in your VNTR region. So hopefully you found this video overview to be helpful. If you have any additional lab questions, please make sure that you ask your teacher before you take your lab quiz.